Hello, welcome. It is March 6th, 2026. We are live in Talktoptera number 5 with Adam Kranz. First, I will pass to Tucker to introduce this session, and then we are looking forward to learning about galls.
Tucker. We're here today speaking with Adam about his work regarding gall and gall formation and interactions between insects and their plant hosts and tissue formation. So Adam and I met, so to speak, via Twitter because he shares really interesting work. And he's also interested in a public science project about gall identification around, I believe, the United States. And so he'll share more about that and his PhD work. So Adam, take it away. Give us maybe a brief introduction about yourself and your PhD and share your work.
Sure. Cool. Well, thank you guys for having me. My name is Adam. I'm currently a second-year PhD student in the Martinson Lab at the University of New Mexico, and I'm the founder, one of the co-founders of gallformers.org, which is a web resource for the identification of plant galls, which I guess leads very well into the first slide here of my talk, which is telling you guys what a gall is. So I think some people have seen them, but very few people actually have a sense of what they're looking at. So galls are, interestingly, novel plant organs, right? So they're made of plant cells expressing plant genes. There's no evidence of gene transfer. It's purely the organism's own genetic code being interpreted in a new way in response to the presence of a specific inducing organism. We call them gall inducers that guide the development of that host tissue. So this life history strategy has evolved many different times independently. Lots of different insects, aphids, psyllids, midges, wasps, mites, moths, beetles, flies, wasps, sawflies, and then there's a variety of other kinds of things that can do this as well, like nematodes, some oomycetes, rust fungi. But it's basically, if you're an herbivore, right, the advantage is kind of obvious because it makes the plant feed you on purpose, right? This is kind of analogous to what humans do with plants, domestication. You see it's when the plant is taking those resources out of storage and deploying them rapidly to develop a whole bunch of new tissue. So think of like the maple syrup flux in the spring, when all of that sugar is coming up from the roots, going toward the leaves to develop new leaves and flowers. That's an opportunity, if you are a herbivorous parasite, to guide the development of new tissue to something that specifically benefits you, as opposed to whatever the host is trying to do for itself. So that means that galls are phenologically, in terms of their timeline, their schedule throughout the year, highly dependent, highly coordinated with the host plant. Right? They're reliant on when that plant is moving resources around. So we end up with galls in two different categories: galls that are produced in the spring during the period when resources are rapidly coming up from the roots, and then over the summer, the plant is accumulating new sugar, new carbon over the course of the year. That also provides an opportunity to grow, but more gradually. So if you look at galls, you'll see different patterns here, right? The galls that we see in the spring tend to be integrated into the leaf, and then the inducer will leave at some point during the spring, whereas the galls that we see over the summer often will drop off the leaf over the fall. But this is just a good opportunity to show some of the variety of the forms that we're talking about here, to give a sense of like how varied the structures that these things are producing are. So these are all of these species here are on grape. These are all different things you might see on a hickory. So the ones on the left are phylloxera, the ones on the right are midge galls. These things are all very small. When you see the pictures of them, it's a bit hard to guess the scale, but we're talking, maybe a centimeter. These are
Adam,
What's up?
I don't mean to interrupt, but I just that picture in the first slide of galls that had those red triangular or conical galls. And this is a general question, but do the larvae induce the plant to produce proteins, in this case, like whatever is forming those colors, that are unique to the galls and aren't expressed by the plant otherwise? Or is it just the pure repurposing of tissues that it's already generating otherwise?
Right. So yeah, exactly. So making a protein that does anything in particular is very hard, evolutionarily, to come up with a sequence that makes sense. So first of all, plants don't hang on to proteins they don't use, right? Like if there's something in the plant genome, it's there for a reason, because natural selection is the thing that keeps that protein. The baseline rate of mutation. So yes, by the fact that we know that they're not inserting new DNA, right? That means that anything you see in the gall cell, any trait, whether that's a pigment, whether that's a hair color, whether that's like a morphology, right? Any of the things you see here, these are all things that are potential within the host plant's genome. And that's like the really remarkable thing to me about this, is that you never see plants do things that look like most of this stuff, right? Or if you do, you'd have to, think kind of out of the box of what kind of plant parts. So maybe they look more like seeds or fruits than they look like most things that you associate with plants most times of the year. But they're being activated in ways, in combinations that are very different from what the plant has evolved them for, right? So, and we'll get into this a little more later, but it's a new way of playing the piano, so to speak, that is the plant genome, right? The plant has a certain set of proteins, and those are the notes. And then the order and proportion in which those notes get played is the tissue, right? Produces the tissue. And so, yeah, they have a lot of control over that. And the goal of my research is basically to figure out how that works. So these are my study species in particular. And these are all on southern live oak, Quercus virginiana. And the remarkable thing about this, right, is that you look at these and you see 8 different galls, right? These things are all pretty much completely distinct from each other. But what it turns out is that these pairs are all each pair is made by a single species. So these species have 2 galls per year. They have 2 generations per year. So these are the oak gall wasps. This is what I study. They're special not only because they are some of the most diverse gall-inducing organisms. So there are, maybe over 1,000 species of them. But they have this really weird life cycle, which is called cyclical parthenogenesis or heterogamy. Their life cycle, so they have a spring generation, which we call the sexual generation, that has males and females in it. Those males and females both emerge in the spring, they mate, and then the females lay eggs. And then those eggs are all female. And they produce a different kind of adult female called the agamic female. They produce a different gall. And these things are so different from each other that historically entomologists were literally not able to recognize they're the same species. And even to this day, the only way we can match them is either through direct observation or through genetics. So yeah, we've got probably over 1,000 of them in North America. The majority, a big proportion of them are still undescribed. We think Mexico is probably the center of diversity in the whole world. So Gall Formers, historically, we started as a website in North America, but we've recently expanded to include the rest of the world. We just don't have our data filled in for anywhere else yet. This was like a couple weeks ago, very recent development. So yeah, we're trying to describe, the sort of the diversity of the continent and build a phylogeny of this group. But just as an example, this is one of my key study species, Melanochnema. This is what that life cycle looks like. So they're kind of an unusual one. They make galls underground on the little shoots, right? They're the suckers of the tree. So right now in Texas, as we speak, we're beginning the period of emergence for these wasps. They come out of these root galls, they crawl up onto the tree where the tree is putting out new spring leaves. They lay eggs in the leaf. So you can see the little oviposition scars here, little dots where those wasps have laid eggs. And then throughout the summer, that egg causes the plant on this lateral leaf vein here. And it's very specific, right? The wasps lay their eggs on the lateral veins, nowhere else. That is the only place this gall can develop. Other galls will develop on the lamina of the leaves, in the midrib, on the petiole, on the stem. They're all very specific. And then over the course of the summer, they grow and grow and grow, and they create this structure, the mature gall, and the wasp will pupate and emerge around December, which is when the plant is putting resources from the leaves down into the roots, right? It's storing them over the winter. And so they lay eggs in the roots and they grow into these sexual generation galls. So that's just an example to contextualize what that means. The interesting thing about that, I have this in here, right? We can think about the fact that not only does the host only have one genome that is making leaves, making fruits, making flowers, and also making both of these two galls, right? But the wasp genome itself, right, is producing multiple different kinds of adults. So that agamic female and the sexual generation female do not, in this case, I guess they don't look that different, but these guys look more different, right? These three wasps are all made by the same genome. So this is the sexual generation female here on the bottom right, and then the upper one is the agamic female. You can imagine how entomologists looking at those would not recognize that those are the same species, right? Because they look like they're part of the same tribe. They have the same overall body plan. A lot of the details are the same. They look like they belong to the same group, but they look very different from each other. And yet those are the same, from the same genome. Here, this is the agamic generation gall. Here's the sexual generation gall. It's this little nub here up in the fresh growth. So the size of them is very different, right? The timing is very different. It's just kind of wild to think about the fact that these are all happening from the same set of genes. So my research is trying to figure out how this happens. Oh, okay, yeah. So this is my central question. I think this is what you guys are interested in primarily, is like how, as a question of molecular developmental biology, how does this happen, right? So we have the assumption when you learn biology that there is, a set of genes in a genome, and that different plants look different because they have different genes, right? And that's true, right? It is genetically inherited. Different plants look different because they express different genes. But then there's this discovery that, well, actually, the plant genome is possible of doing tons and tons and tons of things that were not specifically ever expressed in it, right? This is like this latent potential hidden in that genome that is not accessed until this specific herbivorous parasite shows up and activates it. So like how do they do that? So this is, one way to think about this is to think about, well, actually, organ morphogenesis in general has always been this way, right? Like you have the same genome in leaves, you have the same genome in fruits and flowers. In roots, one genome can do multiple different things. It can be expressed in different ways to produce different tissues. So we know already, right, that there's this gene regulatory network, which is basically how the plant integrates signals from the external environment to figure out its context. So what time of year is it, right? And what's going on around it, and what should it be doing in response to that? What's its history? And then trying to maintain homeostasis, right? Because obviously in order to stay alive, a tissue needs to be homeostatic. It needs to keep enough water, maintain the right salinity, pH, right? All these things to stay alive. But it also needs to change, right? So it stays the same, stays alive, but it develops into some new endpoint. And it does that in response to some external cues. So I have this analogy. I don't know if this is one of these things that I never knew what this was myself. I feel like I put this image on screen and people do not recognize this. Do either of you guys recognize this?
No.
No? Okay. So this is part of how a car works. And basically, it is kind of like a physical computer, so to speak. So it's called a transmission valve body. It is the thing in the car that integrates the signals that you as a driver provide. So I push down the gas pedal, that means I want to go faster. I change the gear, right? That means I want to change the relationship between these different things. I want something to happen. But the car needs to try to do all of that stuff contextually, right? It needs to adjust for the current situation that it's in, the amount of transmission pressure, right? Whatever state it's currently at, how fast the wheels are moving. So this device is designed to integrate what's happening right now with how the driver wants it to change, and to output the correct response, right? That's what this thing is doing, right? And so this maintains, uniform fluid pressure. It has all these physical valves in it that allow it to integrate signals and produce an output. A gene regulatory network is basically doing that same thing. It is taking all of the conditions that are currently dominant in the plant, right? Its whole history and what resources it has available. And then it is integrating these signals, these external signals that might be, developmental hormones or changes in light conditions, right? It's integrating those together to decide what's the appropriate way to change gene expression to produce the next state, right? The output state. And so if you were trying to, interfere in this system to create a gall, I don't know, do you guys have a thought of what you would do? Which of these things would be the obvious place to try and change it?
Dan, you start.
I'll go with Q's for 400.
Okay.
Huh. Yeah, well, because there's no new synthesis of proteins, my initial thought was going to be okay, some sort of new batch of RNA is being injected into the system, and that's what gets it to change. Then we spoke about this briefly offline, Adam. I would think maybe some sort of Levian, electron, ionic gradient might have something to do with it. But since those two options seem to be exhausted, I don't know. So I will go for Q's for 500.
Okay, so the cues is the obvious one, right? Because the cues are what the plant tissue use to figure out what they're supposed to do to change in the future, right? But think about this, right? So in order to produce this wild diversity of output states, right, the thing that you were trying to do to the plant needs to be extremely specific. So we know that it's not where on the plant that larva is interacting with it. It's what species it is, right? So every different species produces a characteristic specific output state. No matter there's some variation in where it shows up on the plant, and you'll still get the same output state. So it's basically there's some information channel that is reliably producing this particular kind of output, and not this one, right? So these are both leaf galls. This one is spherical. It's covered in short stellate hairs, right? It's on lateral leaf veins. This one here is a little egg-shaped, and it has no external spherical layer, but it's covered in these really long hairs. What's the difference between those things? Can you convey that difference in a physical cue? The other thing to think about, right, is that you have to be able to put the plant cells on a trajectory that will keep them developing that way, even after you are no longer directly in contact with them. So that means that the changes have to be something that the cell can inherit over generations as it divides. And you're really small, right? So, in part of this plant, you're inserted as an egg, you hatch as an egg, you're an insect larva that's quite tiny. You don't have the ability to change things like light or pressure, right? You're very limited in the external cues that you can physically possibly change in this environment. So you're super small. You can release chemicals and they can diffuse into your environment, but they have to be highly specific, and they have to be something that are high leverage. So the original hypothesis people assumed was like plant hormones, right? Nobody has been able to show plant hormones reliably produce galls. There is a recent paper that came out that found a particular peptide that can induce, in the presence of hormones, gall-like growth. But again, that's not giving you specific structural information, right? It's not an information-dense channel to get the result that we see here. The other thing to think about, right, is that plant hormones are something that we see in a lot of herbivores. Manipulation of hormones is something that a lot of herbivores do. We see strong changes in the gene regulatory network because, again, any external stimulus the plant responds to, right? It tries to maintain homeostasis, which means that it might shift thousands of genes in response to that, but we still don't see a gall, right? So there's something specific happening here that is well beyond what that kind of information channel of just like a generic growth hormone can control. So the thing that, to me, right, intuitively has to be the answer is that it can't be an external cue. It can't be a change to the current state, right? It's very difficult, again, for a tiny little organism that shows up here to make a change in the ion concentration, right? But what you can do is that you can change the gene regulatory network, right? You can find these high leverage molecular nodes in how the plant is interpreting its environment, right? So these are things like transcription factors or regulatory mRNA or peptide signaling, right? That are like sort of buttons that are hooked up to much larger physical processes that sort of allow you to leverage that effect into the rest of the organism and basically make the plant amplify the signal for you. So that's the broad theoretical idea here, right? And the question is, what does that look like specifically is what I'm trying to figure out. And I don't know the answer to that yet. If you said it was a peptide, I think that would be some kind of signaling peptide might be reasonable. But I don't know. miRNA, so regulatory microRNA, is a tough one as well. Definitely possible, but it's hard to detect transcriptomically. So I guess maybe into talking about methods, or if you guys want to jump in right now and ask some questions.
Well, actually, I was going to ask. So how do you think? How are you going to solve it? Like, you've done a ton of background research, it sounds like, on these specific species and galls in general. So how do you, what are ideas you have for approaching how to test this either in the lab or in the field? So it sounds like
we do
with methods.
Yeah, the first thing we tried to do last year was to find the salivary glands. So as a point of reference, Acerina is a tephritid galling fly, and my lab also studies these guys. And in those flies, there are massive salivary glands, right? Which is sort of the basic assumption is if an insect is producing something like these effector molecules, effectors are anything that the plant would use to control the host phenotype. So whatever they are, they're being expressed presumably in salivary glands because that's going to get pushed out to the host, right? But we looked for these live oak larvae and we took their bodies and tried to find their salivary glands, and we found basically nothing. They're really small. We couldn't find them, and we assumed that must not be the right answer. So the idea was that, right, we were going to take these things, extract all of the RNA. You extract all the RNA and you sequence it, and that tells you what genes they're expressing, right? And so whatever the larva is doing has to be encoded in its RNA, right? It has to be something that is part of its genome that's expressed in a particular part of its body that is in contact with the host. So the problem we went through is that, well, if they're really small, then that suggests that they don't matter that much, right? That if they're tiny, it's a negative signal about their relevance to the organism. And actually, so it turns out there's another organ in the body called the Malpighian tubules, which in insects typically does a function analogous to the kidney. And this guy, Rossig, I found this book over 100 years old that was written in German. I think most people who study this group had never read because it's hard in German. Now with LLMs, I was able to just put this through Gemini, translate it, and get the benefit of all his insights, and tried to figure out, okay, well, maybe it's Malpighian tubules instead. So we went and we sequenced those. Jump ahead.
Wait,
Sorry,
just
Adam,
one. Yeah, I have a question there too.
Please, Daniel.
Yeah, on the translation quote there.
Yeah.
One slide up.
Oh, you want me to go back?
Yeah, I guess well, first just the consideration of Malpighian tubules as vicarious is stunning. But I guess just more anatomically, how would that delivery work? Because the Malpighian tubules are not, I mean, is it like a regurgitation?
So yeah, I mean, this is the reason that led Rossick to this conclusion actually was the observation. So actually, what you can see here, this is the head, right? And you can see the little jaws up here, right? Mouth parts. So this is the larva's head. This is the midgut, the big yellow thing here. That's full of the oak tissue that it has been chewing, right? And swallowing. So that's the midgut. The larval insect, and this is true of lots of insects that live in confined circumstances, they do not pass frass, right? They don't defecate as larvae. So they accumulate everything that they ever eat as larvae into their midgut. It just stays there. They never pass it to the hindgut. The hindgut is, so we call that blind, right? In other words, the midgut has no output. It doesn't go anywhere, which means that the hindgut stays empty, right? There's nothing that ever goes into the hindgut. It turns out that the Malpighian tubules have the possibility of passing things to the hindgut where they're exposed to the anus, right? So in other words, the idea would be that anything expressed in the Malpighian tubules could be excreted out the anus.
So it is a blind gut or not?
It's a blind midgut, yeah. So they're not connected, right? There's the hindgut and the midgut. The midgut, things go into the mouth and they stay in the midgut, and then things could potentially go out the anus from the hindgut, but they're not connected. The things that are coming out the anus are not food. It's not frass.
All right.
So yeah, the remarkable thing about it is that the Malpighian tubules are enormous, right? They take up like the vast majority of the inside of the larva's body that isn't taken up by the gut. And so that again suggests, well, there must be some importance to them. And if they're not producing effectors, I don't know what it is, because I've looked at the transcripts of these things. They're very transcriptionally active. They're enormous. They're expressing a lot of genes. What are they doing? It's not obvious. They're not expressing the kinds of genes that they should be expressing if they were doing like post-digestive, hemolymph processing, the sort of things that a kidney does. They do not look like they're doing that. All of that stuff happens in the gut lining. So, and again, like thinking about ecologically what this insect is doing, it's eating a kind of food that it has controlled to be ideal for it, right? So it has caused the plant to create nutritive tissue that is highly dense in nutrients and lacking in the things that plants usually make to deter herbivores, right? The larva has, manipulated the gene regulatory network to prevent that. So they have this very high quality diet. They don't need to do a lot of detoxification, and yet they have these massive Malpighian tubules that don't seem to be involved in processing their diet. I have no idea what they do. They produce a lot of mucus. If you dissect them, they make these giant stringy, like, you guys have ever eaten natto? It's sort of like that, right? You pull it apart, it's big strands of mucus. I, at this point, do not have a good idea of what these things are for. But we have updated back in favor of the salivary glands, because we did find them and we did sequence them, and they are small, but they make up a really disproportionate percentage of the larva's entire gene expression pattern. And there are specific genes. There are specific genes that I believe to be like signals of the effector group. So there's this one here called endoglucanase Z. Like this is a cellulase. This is a gene that degrades cellulose, right, which is what the plant cell walls are made of. This actually turns out to be lateral gene transfer from bacteria. So one of the subjects of my PhD, one of the 4 chapters I'm writing, my first paper, is going to be about these bacterial gene transfer events. There are actually 3 of them that gave these cell wall degrading enzymes, right? Most wasps do not eat plant material. They cannot eat plant material. And so in order to transition into this lifestyle, the oak gall wasps needed to gain this molecular toolkit that allowed them to open these plant cell walls. And so they got them from bacteria something like 150 million years ago. And that was sort of like the beginning of the diversification of this whole group. This gene is expressed in the adult venom gland, and it's expressed in the larval salivary glands, and it's expressed at like enormous levels. So this is consistently one of the top 10 genes in the entire body of the larva. Even though the salivary glands are really small, they're expressing so much of this that it's one of the top genes in the entire larval body. But it's not, again, information dense enough to control the shape of the gall, right? This is not the thing I'm looking for. So what is the thing I'm looking for? So in this whole transcriptomic profile, you see a lot of genes, right? So for instance, a lot of what I see is this mucus-like group of genes, right? And this is what mucus proteins kind of look like. They don't have fixed structure. So if you do a structural prediction with AlphaFold on them, that's what all these are. These are AlphaFold predictions. You get this disordered stuff, right? And then these end up getting covered in sugars. They get glycosylated, and then that sugar soaks up water, and that like big protein noodle covered in sugar and water, that's what mucus is. So they make lots of these. The salivary gland makes them. The Malpighian tubule makes them. That's a part of this. I don't think that that's the effector, but it does seem to be important for their lifestyle to be able to cover themselves in the right kind of mucus, you know? We get a lot of these particular enzymes that show up again and again in venom. Lots and lots of these proteases. Wasps make tons of proteases. Gall wasps make maybe even more of them. So these are all like sort of known things in wasp venom. This is the one I mentioned earlier, actually. This is the CAP peptide. It was found in aphids, galling aphids in Asia. But there's this particular sequence of 6 amino acids that if you clip it out, you have to cut it out of this protein. Then it becomes this little cleaved peptide. You can use that on its own with auxin and cytokinin to produce this gall-like structure. So this shows up over and over again. We see this in at least 4 different lineages of galling insects. They all express this in their salivary glands. But again, it's not obvious that this carries enough information. Like if you think in terms of how many kinds of peptides you would need to put into the plant to control that structure, right? It can't just be one, because if it was just one and they were all the same, then you'd get the same result every time. And they all seem to really only express this gene once. So there's not a lot of copy diversity of it. So it can't be the thing that controls the structure. This thing may be more promising. This has a known mechanism for, it's called the venom R-like or probable salivary secreted peptide, is a known mechanism to enter the nucleus of plants and to alter the gene expression, gene regulatory network, right? So this potentially an answer to the question, right? Could be the kind of thing that's happening, some kind of protein or peptide that enters the plant cell, potentially enters the nucleus, interferes with the way that the transcription factors and their cofactors interact, and that changes gene expression. That, to me, is like what the story should look like, and this candidate fits that fairly well. But it turns out that this thing is highly expressed in the gut of the insect, and that, to me, is like hard to square with a role as an effector. This thing is my main candidate. I have no idea how it could do anything, but it is very highly expressed in the salivary glands, consistently across species. It has lots of paralogs that are slightly distinct in their structure. So I'm currently in the market for any hints about what this thing does. But you look at it and it's just very nondescript, right? It's just like these alpha helices. It doesn't look like it should be capable of doing anything, but apparently it's important.
Well, are these metaclusters found in other insects doing other things?
So this one particularly is a taxonomically restricted gene group. This is new for the oak gall wasps. Nobody else has anything like it.
What
And that's,
about
again, what I expect. Right?
what about fluorescence or co-localization or co-immunoprecipitation pull down with plants to see what kinds of binding is happening?
Yeah, I mean, once I have locked in on my list of candidates and decide what's worth pushing forward with, those are the kinds of tests that somebody will do. I don't know if that, because that kind of work, especially on non-model plants like oaks, is just so involved, right? I just don't think that it fits into the scope of my PhD work, which is already feeling fairly ambitious at this point.
You got a postdoc lined up, sounds like.
Yeah. So I don't know. Yeah, definitely down the line, like that's the answer, right? But right now, I think my plan is to just publish these as like characterizing a list of candidates, basically, and giving the evidence with them and sort of giving them like a menagerie, bestiary of different kinds of weird peptides that I see. So yeah, we just there's a variety of different kinds of things that don't look like anything that you know. So there are like protein databases, right? You take these and you put these in like Dolly or PDB, AlphaFold, or sorry, FoldSeq, and you get no real hits, right? They're new. They're called taxonomically restricted genes, and they're the kinds of things that allow organisms to do things that haven't been done before, right? So yeah, just lots of weird little peptides like this that show up expressed at really high levels. Don't really know what to make of them.
In your PhD, Adam, will you have a chance to do like gene knockouts, or I don't know if this
No.
works for gall induction, but you actually like mechanically via syringe introduce different cocktails of these peptides to see how the plants react?
Yeah, I mean that is variably possible. We've been thinking about how to do stuff like this, right? Is that you can inject peptides, you can soak things in peptides, you can I don't know, use sort of like little beads that release peptides over time. So stuff like that, potentially possible. You can also, I mean, the problem is that all of the mechanisms for transforming, knocking out genes, introducing genes, right? Anything like RNAi or CRISPR is designed to work on model organisms where you have a population that you can maintain, right? So you'd like to be able to work with them in the lab. And we're working on non-model systems. Oak trees are not good model organisms. We're working on them because the galls are interesting, right? But the actual ability to work with them in the lab is weak. So we've been trying to think about like what is the best model system for this. And I actually did talk to Michael Evan. Actually asked me this, what the best sort of idea for a gall lab model system would be, and I've been thinking about this myself. There's an herb galler, Leposthenes, that is found on all sorts. It's a widely distributed weed. It's called creeping Charlie or ground ivy, Glechema, and it's induced by a cynopoid wasp. It has a similar kind of complex structure to the oak gall wasps. Right? There's like this little larval cell in here, the little nutritive cell, and then the gall grows around it, and it has this like sort of dense hairs on it. And these are same sort of wasp. Right? These are parthenogenic, which means that if you transform one of them, they inherit that, right? So you can make a clonal lineage of something that will inherit whatever traits you give it. So that would be, I think, my candidate for like, what do we do to manipulate this stuff in a lab, but even that would like require a lot of work to set up, and that again is outside the scope of what I am able to accomplish during the span of the PhD. So
Really
Hashtag
cool. You mind if
postdoc.
yeah,
Yeah, you'll never leave. And will you publish it in German? But I was wondering if you've looked at or plan to look at how gene expression changes over time from egg to full-grown larva, and then also how they differ between the two different life cycle versions of this wasp. It's again the all-female version with a different gall, and the male and female.
So unfortunately, part of the reason that these life cycles, a lot of them are incomplete, is that it's hard to find the sexual generation. They're often ephemeral and cryptic, and we either like don't know what they are at all, or we can't find them in large enough numbers to like get that information, or find them soon enough. They're just too small. Like if you look at what we do know here, these are the sexual generation ones are the ones on the bottom here, and they're tiny, right? Except for this one. This is sort of the easier, bigger one. But these guys are all. I discovered this one myself actually, living in Austin a couple years ago. Nobody knew what the other generation of this guy was. These three species we are still missing the sexual generations for. So right now, this species here, Belenocnema, we have samples from both. We have gene expression from both. We have tissue-specific gene expression from the sexual generation. In theory, there should be a gene expression difference between the two, and so hopefully I will be able to identify that. But I have not yet. So it actually turned out that the existing samples I had from that sexual generation were pupa, or they're like late larvae. So they're no longer expressing the genes that I needed to see. So I have my own samples, but they're still waiting to be sequenced right now.
Wild.
I have a
Really
few different questions too.
Sure, please.
So yeah, like just where have people explored if the gall, including or apart from its resource use, is harmful or beneficial for the plant?
So I mean, these are symbioses, right? Symbiosis, word oftenly mistaken to mean mutualism. Symbiosis means two organisms live together, but symbioses over evolutionary time are negotiated, right? Both of the two parties push back and forth, and so we have. I mean, they are parasites by definition, right? They're eating plant material, and in 99% of cases, we have no reason to think that the plant is benefiting in any way. There was a paper that some people wrote about aphids that suggested that the gall was causing like sugar and carbon recycling. There's actually the highest carbon dioxide concentration that any animal has ever been found to tolerate are these aphids that live inside of a gall, because basically if you think what's happening in there, right, they've trapped themselves in a little spaceship, and then they're just exhaling into that all the time. But high carbon dioxide concentrations are good for photosynthesis. But generally speaking, it seems to be the case that these are parasites, right? They're eating plant tissue at the expense of the host. But over evolutionary time, because these relationships co-evolved, they don't cause severe harm to the plant, right? So they're highly localized. So even a big gall like this Distelcaspis here, right? This is maybe 2 centimeters in diameter, right? They don't take very much individually, and they never get to be super highly abundant because they're attacked by tons of natural enemies. So the really annoying thing for me as an entomologist is that I'm trying to rear, for instance, these guys from these galls here, right? So I have people collect hundreds of them, and then I put them in these containers in the lab to try and rear them out. And what I get are other insects, right? I get parasitoids, I get inquilines, things that have killed what I'm looking for and replaced them.
Wow.
So they're massively controlled by natural enemies.
Yeah, it sounds like you chose a tough non-model organism pair to work with. I have a few questions as well, Adam. Just in general about galls, are there any aquatic plant galls, specifically like underwater galls? Because I wasn't aware that fungus would induce galls, for example, and some of the other non-insect taxa that you mentioned. So I'm just curious if there's been any research or understanding about that, underwater galls on seaweed
I have
from.
no idea. So just to show you what the fungal galls typically look like, right? These are rust, and they I can show you my observations. Rust fungi, just produce little fruiting bodies right out of the plant, but sometimes they can cause this kind of dramatic swelling and deformation.
And have you been able to glean any insight on your work from, if there is any foundational work on secretions from these fungal species that induce morphological change in their plants?
Well, so the thing is that, well, yes, these are galls in a sense, right? They're causing a change in the plant growth. They're doing it kind of chaotically. So I probably should actually spend more time looking at how certain kinds of well-studied plant pathogens work, but no, I don't. I mean, they're not doing the same thing, right? They're not causing a predictable organ-like structure to grow the same way every time, like a species-specific organ. So it's a different problem. Like they're doing something different fundamentally.
Yeah. Here's a question. You say you have your collaborators collect hundreds of these galls, and you often are frustrated to see that another insect has inhabited them. At what how, do the other insects also co-opt the teleological or effector machinery to get to the same end goal state of a gall, or do they interrupt the larva's life after the gall is fully formed?
There are actually very rare instances where they do, but typically speaking, they just kill the gall. They just kill it, and sometimes they don't kill it, and they just eat parts of the gall that are unrelated, like outside of the nutritive tissue. But there is one species. So this gall is the original gall here, right? They have this sort of like barrel shape with these little ridges, and they're green, and they secrete honeydew, which is one of these remarkable things, right? If you think about like an oak tree, like oak trees don't make nectar, like they're not bee pollinated, they don't attract pollinators, but multiple species of oak gall wasps cause the oak tissue to secrete honeydew, which is just nectar, right? They're just releasing sugar water in order to attract ants to protect the galls. But anyway, this kind of gall here does get inhabited by what we call an inquiline that turns it into a very different structure, right? That continues the course of development into something almost unrecognizable. But that's a rare thing. And this group here, Synergis, these are actually still closely related to the oak gall wasps. So they have they're part of a galling lineage that became inquilines and then regained, seemingly multiple times, regained the ability to make galls from within someone else's gall. But this is like a rare example. Most of the time, the inquilines just eat the tissue that was already there.
Fascinating. If you took how do I ask this? I'm just curious if in your case with your oak gall wasps, you have the two different, I don't know how you refer to them, generations, the early spring and end of the year. How much, let me ask it like this. How deep is your understanding currently on the differences in what they express? Have you had much luck there, or it's just been even hard to just find both generations, therefore you're restricted more to studying one for now?
Yeah, so almost all the information I have is about the agamic wasp. Like I said, the sexual generation samples I will have, but they're currently in the freezer waiting to be sent off for sequencing.
Let me also ask it. Let me ask it like this too. Why are their galls so different? I mean, because ultimately they're both the same species, and I imagine that their nutritive needs are similar, and their life cycles likely very similar. So why are their galls so different?
It's a great question. So this is actually part of what this little set of slides here is designed to convey. So the spring tissues are different than what you can do over the fall. And so like you have this problem. If you're a spring galler, right, you live on a leaf during the spring and the plant is feeding you, and that's great. You, as soon as the spring is over, you hit this problem, right, which is that the plant is no longer bringing up resources from the roots to feed you. So this supply chain is broken. And so you have to answer that question by doing something, right? I have this funny, I think it's funny. I'm gonna say it's a funny tweet of mine. But mayflies, right? People say that mayflies only live for a day, right? But if they only live for a day, what do they do for the rest of the year? I think it's funny. Anyway, so yeah, an insect has to live the whole year, right? Like these organisms, they induce galls for one part of the season. What are they doing for the rest of the year? So they have to come up with some alternative. One thing you can do is to just like hunker down and wait, right? But the problem with that is that you face attrition, right? You lose individuals. And as a species, it's much more sustainable. You're much more likely to survive if you are getting a second opportunity to boost your population, right? So getting more resources, reproducing again. If you can reproduce twice in 1 year, you're much more likely to survive that year than if you have to wait and just take your chances hiding in some way. So a lot of gall inducers have two-part life cycles. They'll have spring galls where they'll feed on that spring flux of resources, and then they'll do something else. So they'll go to a different part of the plant, or they like the rusts or aphids often go to different plants, right? They'll start on one plant with a spring resource flux, and then they'll go to a second plant with like a summer growth period. That's exactly what the oat gall wasps are doing, right? They're just finding a different place on the plant that has a different resource flow and taking advantage of that as well. So we get 2 niches for gall growth a year. That lets them increase their population. But if you think about it, right, that resource flux in spring is very rapid, and you want to be there for that and increase your population as much as you can. And then in the summer, that resource flow is slower, right? So the plant is photosynthesizing. It's not bringing up a whole bunch of resources all at once. It's gradually accumulating resources. And so you have to be there for longer, which means that you need to create a protected tissue. So if you notice the difference between the sexual generation and the agamete galls, right, these have like defenses on them. They're thick, they're big, they are often very hard, right? This is just like wood, basically, the wasp has covered itself with. And in most cases, they're designed to last a lot longer, right? Some of them will actually fall off of the plant. So some of these are detachable, so they overwinter on the ground in the leaf litter, which can be advantageous because it keeps them covered and moist and stuff. But it's fundamentally a different ecological problem to solve, right? And so that's why they have different responses to that problem, right? They show different morphologies.
Yeah, that's awesome. That's a great answer to the question that I had. Thank you. We have about 20 or 30 minutes left, so I have maybe 2 directions we could take it in, and maybe both. So one is, I know you're involved with popular science or public science project about like an eye naturalist for galls. So maybe you could share work about that. And the second question is, what do you see as like the cutting edge? What's the biotechnology take, if there is one, on gall formation and the sort of chemically induced tissue formation in agriculture or plant science in general?
Okay, well, I can answer one of those questions a lot better than the other. So
take it away.
Yeah, so I started gallformers.org is a website. I think this is more or less still what it looks like. We recently, my co-founder Jeff recently redid the entire website. We got a big NSF grant, $100,000 to pay for improving the website.
God bless, man. Congratulations. That's awesome.
Thank you. Yeah, it's amazing because when I first came up with the idea, well, actually I'm going to tell the story. So Gall Formers is it's a database, right, that just a bunch of volunteers fill in to try and help people study plant galls. We have this ID tool that lets you search for galls by host and then subset it down by all these different traits. So as we saw before, like there's highly distinct sets of traits that are species specific. It's much easier to identify a gall-inducing insect by the gall traits than by the insect traits. And then we have all the primary literature associated with that species that allows you to sort of validate and to see, the chain of evidence and claims that built to that. And I also have this tool to track phenology of when things emerge and when you can collect them for rearing. But fundamentally, right, all those things that we do are cool, but all of this stuff came as an offshoot of iNaturalist, which is, I imagine, a website that you are both familiar with and that many of your viewers are familiar with, right? It's a community science platform for observations of all living organisms around the world. So I was using iNat sort of obsessively during, in 2018, but I was just like a generalist. I was just looking at everything. So I would take pictures of bees and fungi and birds and whatever, trying to identify all the trees. I was trying to get, as many species, as many observations as I could. I was trying to become one of the top observers in Michigan. I bought this book by Charlie Eisman, Tracks and Signs of Insects and Other Invertebrates, and that put me onto all of these weird phenomena that different arthropods do in the environment. So like spider webs and egg sacs and leaf mines, right? And one of those was galls. And I realized that iNaturalist was sort of missing, like nobody was identifying galls, and that there were a lot of resources available that Charlie had sort of listed. And I was constantly like pinging him. I would tag him and say, "Hey Charlie, what is this? What is this?" And eventually he got tired of that, and he just sent me all the papers. And so I started making like a database of what those papers had in them and how to identify all the different galls. And then, there's this computer vision algorithm in iNaturalist. And this was, machine learning back before everyone talked about AI and LLMs. And I found it very satisfying to accumulate enough observations that a species would enter the training data and then would be recognized by users a year later, right? And so I did this for tons of galls. And got them over the line, identified them, put my own observations up. I started collecting databases, catalogs, primary lit papers, making a giant Excel spreadsheet. And it got like unwieldy. Like I couldn't keep it all in my head. I couldn't keep track of it all with an Excel spreadsheet. So I started imagining a database tool for this, and I started basically like begging people for ways to make it exist. And eventually I got lucky and found somebody on the iNat forum who was a web developer who was on sabbatical from his job. And he built the whole thing with me for free. And so that's why I'm glad that now I've been able to get him some money to sort of compensate him for actually doing the work. And now he's, using Claude and very rapidly doing tons and tons of new improvements to the site. And it's very ambitious, and we're sort of, back into a period of excitement. We just expanded to cover the whole world. But yeah, during this period, I started networking with lots of taxonomists and ecologists, and started getting, species described and getting my names in papers, and people are citing us. So it was very cool. And this was how I got sort of into grad school, because I was able to do things that people used, people cared about. Right? I was able to create something that was useful to people, and I'm excited now to be able to make it even more useful. So that's your first question.
Cool. Oh yeah, maybe we could speak on this for a minute. If you do have some insights onto the biotechnology applications of galls, I'd love to hear that as well. But
Yeah.
for two questions for your iNaturalist gall formers project, what's next? And the second question is both for your PhD and for gall formers. I know with that like German paper you translated with Gemini, just maybe briefly speak about how you're leveraging AI to farm, so to speak, all these resources that in the past were unavailable to researchers like yourself.
Yeah, so Gall Formers, what we're doing right now, we recently expanded to cover the whole world, right? So now we have a global map on here. So South America was we originally were just hoping to do Mexico, and then maybe the Central America, and then maybe South America. But eventually I just decided, like, what the hell? Let's just do the whole thing. Because the map tool is the same either way, right? The map tool just loads in this database from some other place. So now, yeah, we have the capacity to cover the whole world, and we just need to add the data. We are Also pulling in plant distributions from existing sources from Plants of the World Online, the Kew Gardens website, and trying to basically think about how can we now, well, first of all, like just the process of shipping updates to the website on a technical level has gotten much faster. Like I'm not involved in that. Jeff does that part of it, but he's been able to move so much faster. Like right now, if I put up a feature request on the GitHub, like before I used to worry like, well, I shouldn't ask for that because that'd be annoying. But now I'm just like, well, he's just going to have Claude, probably just mostly do that independently. And it's not a big deal. So just ask for whatever I want. The cycle is super fast. Things get fixed quickly. It's amazing. But in terms of like what we can do now, one of the things, right, is access all of this literature. So we have all of these, access to all these papers through the Biodiversity Heritage Library. Let's see if I can find a good one here. Biodiversity Heritage Library puts up papers that are public domain, right? Yeah, there we go. So there's hundreds of old scientific papers, and this is what I used to do for gall formers. And I would find these papers, and I would go to them, and I would manually like copy and paste this awkward OCR, and I would paste it into optical character recognition. I would paste it into a program to remove all these extra line breaks, and I would paste it into gall formers one page at a time, and I would manually correct any errors. That's super slow, and I don't have time to do that anymore because I currently consider my PhD research to be more, worth my time. So I have volunteers who are doing some of this stuff, but there's like way too much of it, especially now that we're global, trying to cover the whole world, right? There's just no way to do all of this work individually. But this is now a problem that LLMs can solve. And they can solve it in any language, right? So as long as you like find the papers, what we're working on, one of the things we're working on right now is building a pipeline to take a paper like this and to automatically find all of the entries associated with different calling species, match them up to the current names in our database, and add them to like a format so that we can just pull all this information much more efficiently than we could before. That doesn't exist yet. We're working on it right now. Seems pretty straightforward to implement. You just have to sort of be careful about it. So that's one thing, right? It's just that all of that literature will be able to get entered into the database much quicker than it would have been before.
Yeah, for gall researchers and enthusiasts worldwide. Yeah, and if you had an answer to my other question regarding biotechnology applications, maybe just kind of the pie in the sky frontier thinking on gall tech and gall theory, where are we at?
I have thoughts on this. So this is one of these things, especially when you start writing grants, people are like, why do we care about this? So like gall inducers can do something to the plant we cannot do, right? They can do something to the plant that we don't understand how to do. And so so if you guys think about, for instance, the things that we do, all of the genetically modified plant strains, so everything, all of like the Roundup Ready crops, all that sort of thing, right? All of that technology came from the discovery of how this species, Agrobacterium, works, right? So Agrobacterium has a mechanism to insert genes into a plant, right? That's how it works. Gall inducers do not do that, right? So they have this plasmid, and you can put something and it gets integrated into the plant genome. That's what genetic modification technology is based on. So all of that entire industry came from the discovery of how this one organism made this one very modest change to its host, right? Because Agrobacterium doesn't produce a real gall. It's like a tumor. It's not an organ. It's not structured. It doesn't do any of the kinds of things that gall inducers can do. So in theory, right, like there are two things to this. One of them is that if we understood how a gall inducer is able to control the structure of the plant, we would understand plant development in a way that we do not currently understand. Like there are pathways that control how organs form that we probably aren't even aware of yet. And that are probably being targeted by some of the effector molecules that gall inducers use. and that studying them and finding those would allow us to discover that. And then once we discover them, right, we can, expand what those things can do very rapidly. So it would be good for plant developmental biology, but it would also be good for the laboratory biotech to be able to control, right? Because if you can do what gall wasps do, and you can control how the plant gene regulatory network works, you could test it in all sorts of ways, right? You could find all sorts of specific ways to modify it that would produce like custom organs, basically, that would allow you to study plant development in very convenient and like enlightening ways, hopefully. Again, this is a bit pie in the sky. This is a bit distant from where I'm at right now, but that's like the end point of this in theory, is that you would have access to a way to reprogram plant tissue on the fly to your own desires almost infinitely. If that were true, you could do all sorts of things, right? Creatively, scientifically.
Bioreactor. So sorry to interrupt you, Adam. I'm sorry.
No, please.
I was just going to say, gall is like a mini bioreactor. So you can imagine that if you're able to know what you're going to produce and design the body plan of the gall and its chemical infrastructure, you could make it a mini bioreactor of your choice, producing proteins or sugars of interest, maybe someday even pharmaceuticals, and like the plant becomes the power plant, so to speak, and it produces these little bioreactors for you. It's really exciting, man. Get another grant.
Exactly. Yeah, I don't know. I mean, it sort of depends on what the answer looks like, right? Like, but if you look at the diversity of galls, it seems clear that there is a lot of specificity to whatever mechanism they discovered. So it suggests that that mechanism should be something we can extrapolate beyond what they do. And the advantage of it, right? But this is maybe an advantage or not, depending on your perspective, but it's not inherited. So the changes you make are localized. They're not. So like a lot of people worry with GMOs of genetic material getting copied outside of crop lineages, right? Like resistance genes spreading to weeds or things like that, or having non-target effects, right? But with galls, the mechanism that they use is entirely localized. It has almost no effects outside of the particular part of the plant that you want it to affect. So a very high precision tool.
I think the gall modified organism, GMO 2.0, Agrobacterium 2.0, grow a huge tomato just right there on the side of the stem, or some other sort of absurd combination, custom nutrient ratios.
It's not custom tomatoes anymore, right? You're designing organ. You are an organ designer. So what plant organ could you create if you had full control over the plant's gene regulatory network, right? All of these things, they're like creative, right? You can, that's the amazing thing about this. You look at them and you're like, wow, each of these insects, designed a particular structure to its own needs. Well, what if I could do that, right? What if I could create something that is unlike any fruit that already exists, that was just exactly, something I came up with? I think that, that to me is like the most fun imagining.
Totally. And to use the term you used, and to give it like a Michael Levin twist here, you're able to realize these teleologically determined structures with a tiny bit of input with high leverage. So with just these effector molecules, you're able to get this plant to actually build a coherent, organized structure, or like an actual organ, and not just like have a scar. So that's very like Levian to me. Not to overuse Levin's name here, but the idea that you can co-opt a system and just nudge it in the right direction, and then it fills in the body plan and the work to get towards a final end state, in this case the gall. So yeah, that's very compelling.
Yeah, I think that's exactly right. Like, I mean, the word you used, teleological, is precisely the right one here, because for me, people, we like to argue about this, scientists, nerds can be very pedantic, but like this claim that galls are novel plant organs, in my mind, that excludes certain kinds of, responses. Like that rust fungus is borderline, the Agrobacterium is borderline, because they're not causing the plant to pursue a specific endpoint, right? Like when you're developing as a fetus, the human fetus, right? Your tissue has a goal in mind of a particular kind of structure, like layers of tissues, gaps between the tissues that are supposed to happen, right? And galls do the same thing, right? They're able to, one of the more remarkable ones, I think this is just sort of like the most illustrative of way to think about this. If you produce a tumor cells do not, they do not die on purpose, right? Like when your hand grows, you have gaps between your fingers because some of that tissue underwent programmed cell death in particular ways, right? One of the really remarkable oat galls is this roly-poly gall, right? Where the cell was initially suspended in the middle of this gall by a bunch of fibers. All of those tissues have disappeared, right? Those plant cells underwent programmed cell death in order to destroy that tissue entirely, which causes the nutritive cell where the wasp is to fall. And that makes it loose, which means that it's harder for the parasitoids to find it, right? Because when they put their ovipositor in there, they just push it around, right? It's loose. It's not suspended in place, so they can't localize it. So this is a defense for the wasp, but that's the goal, right? There is a specific endpoint structure that includes some tissues that are present, some that are absent, that is functional. It's to create this like defense against parasitoids that can only happen if the wasp is able to encode. And again, this kind of structure, oak trees do not do this, right? Like you will not look at an oak tree and find like a rattling, part of the plant that's detached from everything else. Like that is something that this wasp has made that plant do. It would not normally be able to do.
It's like first year PhD, how does the gall build what it builds? Second year PhD, how does it not build what it doesn't?
Well, I mean, we do have a sense of what those programs look like. Like there are actually, I mean, so it is true that plants generally don't completely destroy their tissues. But if you take a step back and think, what is a tree, right? A tree is constructed out of dead plant cells, right? So plant cells do have programs to die on purpose. And mostly the plant uses that in order to create the xylem tissue that carries water and the wood that actually holds the structure of the plant. But this thing where they actually die and disappear completely is a little bit unusual.
Yeah, like a proper organ with not only morphological differences, but morphological differences over time and all the genotypic expressions that go along with that. And the fact that it all comes from some relatively small cocktail of chemicals and interactions from these tiny little eggs is unbelievable, man. It gives me goosebumps. It gives me gall bumps.
Yeah, I love it.
Yeah,
I'm really excited. I'm glad to be able to study this.
Yep.
Feel
free to. Oh, sorry.
Sorry, Brett. I was just gonna say, who's your PhD advisor and what's your lab, and then Dan will help us bring it home.
So my advisor is Ellen Martin. Let's see. She have a. This is our lab. Ellen Martin's lab.
Awesome. Yeah, we'll share that in the video notes. And then Dan, bring it home.
Yeah. Feel free to make any last comments. Otherwise, this has been a great presentation, and it's an exciting research direction.
Awesome. Yeah. Thank you guys for having me. It's been a pleasure.
Adam, I'd just like to ask one departing question. Can we participate on gallformers.org? Can people, like on iNaturalist, can we take photos and submit them?
Yeah. So iNaturalist is sort of our, recruitment mechanism, right? As if we notice some user who keeps posting a lot of galls, we'll say, hey, you want to join our community? We have connections with all these scientists. We can do things that are exciting and interesting. We have a Discord server that if people are interested, you can just join that and then get involved that way. But yeah, iNaturalist is the place if you want to post observations, right? And then just use the website to try and learn about what you find, and or you can just get in touch with us and tag us on
Great.
iNaturalist.
Can't wait. Well, thanks again, man. This was fantastic. Maybe in a year or two, we'll check back in on the progress on your PhD and double back. By then, you might work for DARPA and you won't be able to talk to us. But
Yeah, I know it's funny because you ask these questions at the beginning, like you're gonna tell me how this works, and I'm like, yeah, I would love to tell you how this works. But that's the kind of the beautiful thing about being a biologist is that everything is really, really complicated. And so you start this project with these big ideas of what you're going to accomplish, and then you encounter the reality of the situation, and it's like beautiful, right? And it's so cool and so satisfying, especially now. So I can make a lot of progress with Claude doing my analysis, but it's just
awesome.
it's just tough, right? It's slow going, so we'll see.
Well, man, yeah, we appreciate the important work you're doing, enlightening us and the scientific community on this awesome phenomenon. So
Yep.
we'll stay tuned.
Thank you, Adam.
Wonderful.
Thank you, Tucker. Thank you, Mills8102 in the chat. Farewell.
Let's go. Talk to
Bye.
you guys soon. Have a good one, Adam. Bye.